On behalf of Cambridge Health Tech Institute's Global Web Symposia Series and our sponsor, Demetra AgBio Incorporated, I'd like to welcome you to Cas-CLOVER, the clean alternative to CRISPR-Cas9 and piggyBac for cell line development and engineering. My name is Elizabeth Lamb, and I'm the host and moderator for today's event. Now I would like to introduce our presenters for today. First is Kayla Bean Ph.D., Research Scientist, Discovery Research with Elanco Animal Health. Our second presenter is Jack Crawford, CEO of Demetra AgBio Incorporated. Now I'll turn the webinar over to our presenters.
I would like to begin today by thanking Cambridge Innovation Institute for holding this webinar. I would also like to thank Demetra and Jack for the opportunity to present this work today. It has been a pleasure to work with Demetra and their team, and I look forward to continuing our collaboration on this work in the future. Again, I am Kayla Bean, a postdoctoral research scientist at Elanco working as a cell line engineer in discovery biology. Today, I will be speaking to you about how we at Elanco have utilized Demetra's technologies of Cas-CLOVER and Piggyback Transposases for our work in cell line development and engineering. I would just like to begin by giving an overview about what I'll be speaking about today. I will begin by speaking about Elanco. I understand that most of you are not familiar with Elanco, so I will start by giving a background on our company's motto, our history, and animal health in general. Then I will give an introduction into Demetra and give an overview of the technologies that they provide, the Cas-CLOVER gene editing system as well as the Piggyback Transposase system, and how they can be used in industrial processes for improvements in areas like cell line development. Here at Elanco, we consider ourselves a purpose-driven company. We endeavor to benefit animals and their lives. We use the motto, making their lives better makes life better. And we understand that now, more than ever, due to the pandemic, people need pets and companionship as well as a stable, quality food source. Elanco was originally founded as a division of Eli Lilly, with our company name meaning Eli Lilly and Company. Elanco has over 10,000 employees and growing due to our recent acquisition of Bayer Animal Health. We have products sold in 90 countries with more than 200 brands. Our company is now positioned in the top 3 animal health companies globally, and we continue to strive to empower farmers, veterinarians, and pet owners to have the tools and products needed to allow animals to live healthy lives. This is a timeline that depicts Elanco's journey as a company. When the company began in 1954, it had an agricultural focus, mainly on crop science. In the 1990s, Elanco moved its sole focus to animals, acquiring livestock and companion animal vaccines in the 2000s. In 2018, Elanco broke off from Eli Lilly and became an independent, publicly traded company where we can dedicate our time to the well-being of animals. And in 2020, we completed our acquisition of Bayer Animal Health to continue growing our company. When it comes to animal health, really before 2000, anything termed biologics meant the use of a vaccine. However, that terminology has shifted. Monoclonal antibody therapies are now a part of the animal health market. A blockbuster drug in the animal health space would be considered to be a therapeutic that makes $100 million, which is about 1/10 of the revenue of what a human blockbuster drug would be. The first example of this type of drug is called Cytopoint, which targets the IL-31 to block itch signals in atopic dermatitis. This drug has now become a blockbuster with a revenue of over $150 million. Trends like this in dermatitis, pain, and oncology are areas of interest within animal health, with a large focus in companion animal. Also, it is important to note that while animal health regulatory perspectives are different, with 2 paths instead of just 1, the standards are very similar to that of human. In regards to our collaborators, Demetra has 2 technologies that can be of use in developing therapeutics. Demetra is the exclusive licensor of Cas-CLOVER and piggyback transposases. These technologies can be utilized for gene editing in fields such as agriculture, pharmaceutical bioprocessing, and in the industrial biotech space. Demetra has a single license for each of these technologies, which includes the freedom to operate in research as well as commercial areas. After my talk, a Demetra representative will discuss licensing information further. The first Demetra technology I will be speaking about today is the Cas-CLOVER system. Cas-CLOVER is a hybrid gene editing system. This system utilizes a nuclease inactivated Cas9 protein that is fused to a Clo51 endonuclease. Here, the dead Cas9 is purely used as a fusion protein and does not have the ability to cut DNA due to its mutation. The Clo51 endonuclease actually cuts the DNA. However, this cleavage depends on the dimerization of the Clo51. Here you can see a depiction of the complex that forms, which I will go into more detail on shortly. Cas-CLOVER has several benefits to how the system works. The flexibility of the guide RNA design makes the system easy to use and gives a high specificity due to the use of the two guide RNAs. It is very efficient due to the ability of the dead Cas9 to recognize the correct area of DNA, and since the Clo51 nuclease can only cut when dimerized, the system has high fidelity. Just a note on my experience using Cas-CLOVER and the uniqueness of the system with the PAMs on the outside. When designing the guide RNAs, there can be a difference than with other systems. The reverse complement of the left guide RNA needs to be ordered, but this uniqueness also adds to the specificity of the system. In this image, you can see how the Cas-CLOVER system works in the complex forms. The dead Cas9 with the Clo51 mRNA would initially be transfected into the cells and given time to translate. Usually 6 to 8 hours is a sufficient amount of time before a second transfection would incorporate the guide RNAs into the cells. Once the guide RNAs are present, they will form a complex with the translated dead Cas9 Clo51, where the dead Cas9 acts as a carrier protein to recognize and bring the complex to the correct area. When the complex has fully formed and dimerized, the DNA will be cut. Once the DNA is cut for either a knock-in or knockout, DNA repair within the cell will occur. At this point, you can select for the phenotype or expression of interest. A clear benefit to the Cas-CLOVER system is the flexibility of the spacer links in the guide RNA design. As you can see, using spacer regions of between 16 to 40 base pairs can yield high percentage knockouts. In XB293 CHO cells as well as Dracat cells, studies were performed to assess the spacer regions that would give the best results. High gene knockout efficiencies were seen in a number of different spacer links, proving the flexibility in that cell type. Some spacer links are less than optimal, but this can vary by cell type, target, and should be determined empirically. Cas-CLOVER has been used to knock out the glutamine synthase gene in adherent CHO cells. The Cas-CLOVER technology can have an increased efficiency when transfected into the cells more than once. Here you can see how the knockout frequency increased upon a second transfection without increasing any off-target risks. The next few slides will show how Cas-CLOVER can be utilized for a glutamine synthase gene knockout in suspension CHO cells. Suspension CHO cells are predominantly used in industrial processes, and for this experiment, a serum-free suspension CHO-K1 line was developed in-house and utilized. Once the Cas-CLOVER edits were made, the efficiency of the knockout was assessed using a T7E1 cleavage assay. For those of you who are unfamiliar with this type of assay, the cells are amplified in a sequence-specific region, then denatured and reannealed where a heteroduplex formation should occur. Areas where a CRISPR edit have occurred will have a mismatch in this heteroduplex area. Those areas will then be targeted using digestion with a T7 endonuclease and ran out on a gel. The digest should reveal multiple fragments reflecting the heteroduplex mismatches to confirm a knockout of the gene in those cell populations. Cleaved fragments are proportional to the individual genomes where there was an edit. Here's an example of the result from a T7E1 cleavage assay on a glutamine synthase knockout in suspension CHO. In this experiment, 17 guide RNAs were designed to target knockouts in exon 1 and exon 5. 4 pairs in exon 1 you can see here to the left, 2 of the exon 5 pairs you can see here on the right. Continuing to the next slide, you can see the remaining pairs that were in exon 5. And these knockouts were evaluated using that T7E1 cleavage assay. When the band intensities were calculated, the overall edit efficiencies ranged from 10 to 40% knockouts within the CHO suspension cells. Other ongoing projects in our Demetra collaboration include utilizing Cas-CLOVER system for sequence-specific targeted knock-ins. The location of this knock-in will utilize what we call safe harbor loci regions in CHO cells. These regions have been targeted due to the ability to achieve high gene expression without disrupting surrounding genes. For this type of work, we will be designing and screening guide pairs for cutting efficiency, as well as building donor plasmids with homology arms to flank the gene of interest. Our initial work uses GFP as a proof of concept, and we are optimizing and validating protocols. We do have some promising GFP positive experiments, but the results are too preliminary to discuss today. However, some Demetra collaborators will be present at the end of this talk to talk more about some experiments that are ongoing. Switching gears, I would like to take a moment to discuss our use of Piggyback Transposase technology by Demetra, coupled with our Berkeley Lights Beacon platform. I'll give more insight in how we utilize Piggyback Transposase in our cell line development processes and outline how we achieve single-cell cloning, including our Beacon platform, to have higher producing stable pools as well as clonal cell lines. Piggyback Transposase works in conjunction with a transposon vector harboring your gene of interest. The transposase recognizes ITR regions seen in the image. The transposase and the transposon vector are co-transfected into the cell. The transposase is used to cut and paste a fully intact cassette at TTAA sites using random integration. Due to the fact that transposases prefer active chromatin, high expression is usually seen. This type of technology is also beneficial because it is a non-viral and cost-effective technique with the ability to have high genetic stability with small or large gene integrations. At Elanco, we have generated our own plasmid systems utilizing the Piggybac ITRs. This highlights the flexibility of the Piggybac system to be dropped into existing expression platforms by simply adding Piggybac ITRs. At Elanco, we have performed extensive transfection and selection methods using Piggyback Transposase for integration. We have successfully tested a wide variety of transfection reagents and systems such as Freestyle Max and the Neon transfection system. We have successfully generated high-producing pools with multiple selection methods, such as the MSX selection for a glutamine synthase knockout CHO line, and antibiotic selection methods, such as puromycin, which can generally be done in a shorter timeline. By and large, we are integrating and producing canine monoclonal antibodies here at Elanco, where we routinely see 1 to 3 grams per liter production in our stable pools, depending on the type of molecule being produced. For our single-cell cloning procedures at Elanco, we use a Beacon platform from Berkeley Lights. This platform, seen here, which is about the size of a typical vending machine, houses an entire microfluidic system for single-cell cloning. Also, the Beacon platform allows for assaying of these cells on chip. Those chips can be seen to the left. The Beacon technology utilizes an optoelectric pulse to pin single cells into individual areas of the chip, which I will show in more detail in a moment. Here you can see where the chips are housed in the biosafety cabinet-like area where positive pressure is utilized, as well as a perfusion system to culture cells on chip. To give a better view of this chip, you can see here what a pen looks like. Each chip has 1,768 pens, with each pen having a volume of 1.7 nanoliters. 4 chips can be ran simultaneously during one run. The platform can be used to assess monoclonality by ensuring single-cell loading, and also study the growth of those cells on chip. In image 1, you can see single cells from your stable pool populations have been sorted and individually penned. In image 2, you can see those cells culturing and the different growth rates between each of the pens to help assess which cells are growing faster than others. In image 3, you can see how the Beacon platform gives the ability to assess target protein production on chip. The signal being generated from a fluorescent-based reagent is proportional to the antibody or target protein being generated. In image 4, you can see an export of the top clones with the highest assay scores at day 5. The ability to export your high-producing cells within the chip populations really helps to pinpoint those cells of interest and increase the efficiency of your cell line development processes. Here is just another Beacon assay image depicting all 1,768 pens. You can see the fluorescence achieved across the entire chip during the assay. The more green the pen is, the higher the target protein production in that monoclonal cell line. Here you can see the top clones are highlighted in red boxes, and those would be targeted for export and expansion to be used in future studies. Here are some graphs showing the distribution of empty pens on the left to the colony pens on the right. Quantitative scores here show the difference between background and baseline levels to that of producing cell pens, and this representation of the top assay scores can be seen in the top right graph. Pinpointing the top clones on chip can help to lower the number of monoclonal cell lines that need to be screened, saving time and resources. When coupled with the piggyback technology, the number of high-producing monoclonal cell lines can be increased due to the possibility of high copy number insertion with the transposase. This allows us to identify the high-producing clones from multiple parallel pools simultaneously without sacrificing additional time and reagents. Here's an example of typical Cld workflows from transfection to master banks using both the normal Cld process as well as the process using a Berkeley Lights Beacon platform. A normal process can take 8 to 12 weeks just to expand cultures and assay for high-producing cells. However, as you can see with the Beacon platform, this timeline is accelerated in just 5 days to do the same amount of work. Coupled with the piggyback integration, there's an increased confidence in our stable pools, which will give us a larger number of cell lines to screen to hopefully pinpoint a higher frequency of cells that are producing our target protein at a faster rate. This not only allows us to accelerate our Cld workflow, but to run the entire process with just one individual. Just to kind of give a summary about what you've heard today, we've spoken about the Cas-CLOVER systems, how they were versatile and easy to use and have very high fidelity when gene editing. Specifically in bioproduction cell lines like the suspension CHO K1 lines that you've seen examples of here today. Piggyback transposases can also be used for rapid, high productivity, stable integration of intact gene expression cassettes in a wide variety of cell types. And together, these tools can be paired with other technologies like the Beacon platform to form a foundation of a robust cell line engineering and development workflow. At this time, I would like to acknowledge all of those who've been a part of this collaboration. At Demetra, Kyle Rector and David Norman. From Hera, I would like to acknowledge Valeria Steffi and Daniel Machado. At Elanco, I would like to highlight Alan Spear, as well as our other colleagues John Snyder, Crystal Paulson, and Heather Ringham. And of course, our colleagues at Berkeley Lights. Thank you so much for taking the time to listen to me speak today. And at this time, I'd like to turn over the presentation to Jack Crawford at Demetra to give an update on some of their research before we have our Q&A session. Again, thank you.
Thank you, Kayla, for a wonderful presentation on your work with the gene editing technologies in CHO for cell line development at Elanco. I'm Jack Crawford, the CEO of Demetra AgBio. I'm going to go over a couple of the more recent studies that we're doing with Cas-CLOVER in different cell lines, particularly in knocking in genes of interest, which will be very important for cell line development using Cas-CLOVER. First, the study that we did was to knock in a proof of concept GFP and puromycin at the AAVS1 locus, which is a safe harbor site in the human genome in HEC293 cells. Now below, you'll see two different time points, 5 days post-puromycin selection and 12 days post-puromycin selection. The proof of concept donor vectors that we have and used contain piggybac inverted terminal repeats flanking both the GFP and puromycin. What you can see is we get decent amount of GFP expression 5 days post-puro selection, and then much more robust GFP expression 12 days post-puromycin selection. That's because the puromycin will enrich cells for targeted knock-in of GFP or any other gene knock-in that you would like. We did complete sequence verification of the targeted integration using the ITRs and also flanking regions of the AAVS genomic sequence. So we did confirm that this was accurately targeted into the genome at AAVS1, not a random integration. And then one could, down the line, use a special piggyback transposase that will excise selectable markers that we call excision-only piggybac, or PBX, and that can be used to remove selectable markers as needed. Secondly, what we're doing, and we're still in the process, these data are hot off the press, to use Cas-CLOVER to target at the safe harbor site H11 in CHO cells. And we had basically the same proof of concept vectors with piggybac ITRs, GFP, and puromycin. Instead, the homologous recombination is at the H11 site. And you can see 5 hours now, just hours, not days, after transfection, we don't see really any GFP expression, but then 72 hours post-transfection, we see very robust GFP expression. Now, I guess this could still be transient expression. However, our next steps are going to be to confirm that we do get robust targeted integration at that site in CHO. So we're going to be doing puromycin selection to enrich the cells for targeted integration, and then it can be removed again with PBX. And our next steps with the system for cell line development in general are going to be to fully optimize cutting efficiencies at the H11 safe harbor locus. And we can do other safe harbor locuses as the client would want. We expect once we do fully optimize this to get cutting efficiencies up in the 80 plus range. We do see that with Cas-CLOVER in human T cells, very difficult to edit human T cells. We also see that with Cas-CLOVER in yeast and in plants. Very robust data showing that there's a broad use of Cas-CLOVER for gene editing. So to wrap it up, from Demetra, we are the exclusive licensor of Cas-CLOVER and piggyBac for these fields of use, including pharmaceutical bioprocessing, agriculture and engineering, yeast and other organisms for industrial biotechnology. One caveat to our exclusivity is the use of piggyBac only for human biotherapeutic production in cell lines. That technology was sold to Lonza. So outside of that, we do own all these fields of use. Every field of use we do own also for Cas-CLOVER. So that Lonza agreement is just for piggyBac and just for humans. So animal health bioproduction is something that we work on. The interesting and most advantageous thing, in addition to all these technology advantages that we've shown, is that we have full freedom to operate with a single license for research and commercial use. We have issued IP both in Europe and in the US, and we're going after IP elsewhere in Japan, Asia, China, and other locations. We are providing gene editing services for CHO, HEC293s, and other cell lines, as well as agricultural and yeast-based systems. And so all of these are available to you if you partner with Demetra, like Elanco did with great success. Now we're going to do our live Q&A. And again, I'm Jack Crawford, the CEO, and this is my contact information. Thank you for joining us.
Thank you very much, Kayla and Jack. We do have some questions that have come in. Our first one is for you, Jack. And that question is, what are the typical licensing structures for commercial use for Cas-CLOVER?
Thank you for the question, and thank you, Kayla and Elizabeth. Thank you, Kayla, for the presentation, and Elizabeth for organizing this, and also to the attendees. So we're quite flexible with our licensing, although we do have typical structures that we prefer. One thing I want to go back to was on the intellectual property, and that is one of the key advantages, especially for commercial use in bioproduction, compared to CRISPR-Cas9. And our tagline for Cas-CLOVER is it's the clean alternative to CRISPR-Cas9. And so that refers to the technical aspects where Cas-CLOVER has been shown to have fewer, very, very low towards the undetectable mark of off-target mutagenesis because of its dimeric nature in compared to CRISPR-Cas9, which has known pretty high off-targets, especially in CHO. But it's also much cleaner technology on the intellectual property side, where CRISPR-Cas9 is entangled and messy. Different patents need to be licensed in different areas, the US and Europe, and other issues. But to get to the licensing question, we have three structures. One is mainly for big pharmaceutical companies. And before I get into this, let me mention, we try our best and are very successful to eliminate the idea of having royalties on the cell line or the product. So for big pharma, we do a single upfront fee. That's it. No milestones, no royalties at all. And then for medium size and small pharmaceutical biotech, as well as big contract manufacturing organizations, we do a little bit smaller upfront fee, and with way downstream commercialization milestones. So we still do not have any royalties there. And then with small pharmaceutical biotech companies and small CDMOs, what we do is we do a yearly fee, which is even smaller, plus commercialization milestones. So those are our preferred structures.
All right, thank you. This question is for Kyle, and it's from Abdallah. Have you tried the piggyback system in plants?
So to date, Demetra has not used the piggyback system in plants. We have used the Cas-CLOVER system, and I will get back to piggyback in just 1 second. We have successfully used Cas-CLOVER in plants, in tobacco plants, and Arabidopsis plants. It has comparable efficiencies to that of Cas9. The piggyback system has been shown to be used in rice. It was used to knock in a gene and then with a selection marker. And because of its footprint-free removal, they used it. They later introduced the transposase to remove the selection marker in a footprint-free fashion. But we do intend to use it for a couple of projects in tobacco to introduce stable high copy numbers of particular genes. Yes.
Thank you very much, Kyle. And now a question from one of our attendees for you, Kayla. Could you share with us what suspension CHO cell line you used to create the knockout cell lines?
Hi. Again, I just want to start by thanking Jack and Elizabeth for allowing me to do the webinar today and all the attendees. In regards to the cell line, we obtained a CHO-K1 cell line from ATCC, and we adapted and selected for it in-house to come up with a serum-free suspension CHO line, and that's what we used for this work.
Excellent. Thank you. This question from AJ is for you, Jack. Could you advise on the pros and cons of Cas-CLOVER and CRISPR-Cas9 in gene editing? Are there any significant economic differences?
Sure, it's a great question. The advantages of Cas-CLOVER also have some, as Kayla mentioned in her presentation, require a little bit more upfront design work and implementation, possibly optimization in your cell line. So it's a dimeric system. So you have 2 guide RNAs that need to be designed, and these are relatively easily designed, and we have protocols for both the design component as well as editing in CHO cells and HEC293 cells and other systems. But you need to design 2 guide RNAs and then produce those 2 guide RNAs. It's not a big economic expense. As Kayla showed in the suspension cell line, we were able to design and implement 15 plus pairs of guide RNAs for Cas-CLOVER at a single gene. So the flexibility of what's called the spacer region allows for flexible targeting. So Cas9 is a single guided RNA. That is a technical advantage because it's easier to design and produce. Not much easier in my opinion or our experience, but it is. And the downfall of that, though, versus Cas-CLOVER is it's well documented that you're going to get cutting all over the genome. Your target site with Cas9, CRISPR-Cas9, you'll get cutting, but then you're going to get significant off-target cutting. And this is extremely important for cell therapy and other applications where the cell might be going into a human being, but it's also important for CHO cell line development, because you may be creating phenotypes in your cells that are not desirable without even knowing it if you use Cas-CLOVER. So I would say that's a technical, or if you use CRISPR-Cas9, you're not going to have that issue with Cas-CLOVER. So those are the technical differences. Really, once the system's optimized in your cell, the cutting efficiencies are pretty much the same, and you don't have to worry about the off-target effects. And we've got other data showing the lack of off-target effects. Now, on the licensing and intellectual property, as I mentioned it earlier, that's going to be really where you save by working with us with Cas-CLOVER. We've got a very clean single license system, issued IP, whereas it's extremely messy. You might have to get multiple licenses from multiple groups with Cas9, CRISPR-Cas9, and even then it's unclear whether that IP is going to be enforceable with a downstream product. So those are the key advantages. Thank you.
Thank you very much. Our next question, since Cas-CLOVER is dimeric, do you get different sized indels than single-guided CRISPR-Cas9?
Kyle, why don't you take that one?
Yeah, so what we see in this system is that relative to Cas9, which you normally see indels that range between 1 base pairs to say 3 base pairs. That's sort of the average that we normally observe. With Cas-CLOVER, you see indels that range from about 8 base pairs to 40 or 50 base pairs between the two guides. So you get a relatively large deletion, which makes it easier in the long run to actually detect cells that have the indel. So you don't necessarily have to do a lot of complicated assays. You can simply just do PCR and run it on a relatively high percentage gel, agarose gel or polyacrylamide gel, and actually detect those indels much more frequently. And we suspect that these indels actually will also enhance the knock-in efficiency, and this is something that we're actually testing right now.
Excellent, thank you. Ben asks, what studies have been done to ensure Cas-CLOVER has fewer off-targets?
So our sister company, Poseida, has actually done several studies using NGS, looking, and Kayla's presentation showed this, looking at different sites in the genome that actually have homology similar to that to the target sites in TCR alpha and beta 2 microglobin. And so they looked at these sites, and they looked at the left guide sites, sites of homology, right guide sites, and also sites where you might have 2 left guides together, a site that has homology like that, or 2 right guide sites. And actually what they see when they do deep sequencing, deep amplicon sequencing, is that they actually see no off-target effects. And they've done this multiple times in Jurkat cells and T cells from human samples.
All right, thank you. Denise asks, have you tried gene editing CHO cells with Cas-CLOVER RNPs?
So that experiment is actually in our queue. So we're currently producing the Cas-CLOVER protein. This should be finished end of January, and those experiments we'll embark on those within a month or so. Yeah.
Excellent, thank you. Our next question, how do you design gRNAs? Is this proprietary, or can you use open source software that's compatible with Cas9 nickase systems?
So the softwares that we use are open source softwares. These would include eCRISP or ZFit or Synthago websites where you can actually use the software that they designed for the Cas9 nickase systems, and it will design guide pairings that are compatible with the Cas-CLOVER system with gap distances that are appropriate for Cas-CLOVER, which are between 15 base pairs and 30 to 40 base pairs.
All right, thank you, Kyle. I think this question is for you as well. This one is from Chris. As Cas-CLOVER requires 2 gRNA, what is the flexibility of their proximity to each other without affecting efficiency? And have you estimated the proportion of the genome that is available to editing as a result of PAM availability?
So the first question, the flexibility, like we said, it ranges from 15 base pairs to 40 base pairs. We comfortably work between 15 base pairs and 30 base pairs. And if we do not identify appropriate guides that lie within that space or region between the two guides, then we'll further extend the space or region to look outside of that between 30 and 40 base pairs. But it's the frequency of guide sites are certainly easier to find once you extend it out to those larger space or distances. And we have not encountered a gene to this point that we cannot target. In addition to that, you can look outside of the exons just a bit if you're working in CHO cells to kind of include regions that lay outside of the exon, which you can't really do with Cas9. So you can design one guide that lays about 20 base pairs to 30 base pairs outside of the exon, and the deletions are usually so large that you'll encounter those occurring within the exon region.
All right, thank you. A question from Michael. Does the guide RNA for Clover Cas still need CR-RNA and tracer RNA?
Yes, so we use single-guided RNAs for all of our experiments that we do internally, but just like Cas9, if you prefer to use the tracer RNA system, cRNA system, it is definitely compatible with that.
All right, and we do have one last question that's popped up here. How do editing efficiencies compare to Cas9?
So in our hands, the editing efficiencies are almost identical to that of Cas9. So you don't necessarily lose on efficiency in your experimentation.
All right, thank you so much. That appears to be all of the questions we've received. So I'd like to very much thank our presenters for today, Kayla Bean and Jack Crawford. I'd like to thank our sponsors, Demetra Ag Bio Incorporated, for sponsoring today's event. Finally, I'd like to thank those of you who came and spent this time with us. We know you're being pulled many directions in these strange times, and we're very grateful that you chose to spend this time with us. So on behalf of Cambridge Health Tech Institute's Global Web Symposia series, thank you all so very much, and have a great rest of your day. Bye-bye.